The law of independent assortment has to do with metaphase 1 of meiosis. So here we're seeing a cell that happens to be in metaphase 1, and what happens during metaphase 1 is the tetrads line up. Well, what the law of independent assortment refers to is that each tetrad is going to line up independent of the other tetrads, and this is going to affect the end results that we get with meiosis. So this will affect which chromosomes end up in the different gametes. So in other words, how this one lines up does have no effect on this one, and again, no effect on this one at all. So it is possible that we get them lined up like you see in this picture, where all the blue happen to be on one side and all the red happen to be on another. But it's possible that they could line up differently so that you get a mixture of red and blue chromosomes on the two sides when they do line up. So notice here that with independent assortment, if we were applying this to genetic crosses, the genes assort independently when they are on different chromosomes. However, that's not always going to be the case. So when we think about a particular chromosome, on a single chromosome, there can be literally hundreds or close to 1,000 or more genes that are located there. So if we're just talking about two random genes that we select, it's very possible they're on the same chromosome. So if we have genes that are located on the same chromosome, so if this is my chromosome and we wanted to track, say, gene A, look it up here, gene B, gene C, gene D, all of these, since they're on the same chromosome, we will refer to them as linked genes. So that's the relationship they have with each other. And the reason we're calling them linked genes is because they are on the same piece of DNA. And for these, they do not assort independently, and that means that they tend to stick together, or in other words, they tend to be inherited as a unit, is another way to say it. So for example, where that big A gene ends up ultimately in a gamete, it's very probable that the big B is going to end up in that exact same gamete. So if we talk about this a little bit more, when you do have genes that are located on the same chromosome, they can still be separated during the meiosis process. So if we were tracking multiple genes on here, and I'm just going to write an A and a B, if we think about what might happen if we had crossing over, one chromosome might have two capital A's on it and two capital B's, and then if we had a homologous chromosome there that we might have crossing over with, if I draw that next to it, on this one maybe we have two lowercase a's and two lowercase b's. Well, suppose that we do get breaking of this chromosome and we're going to get crossing over. Now, if crossing over takes place and we actually get crossing over between the A and the B, so notice that the crossover is specifically between the A and the B, which are the two genes that we are tracking in this particular example. If the crossover is right there, then you do get separation of those. So you would have a swapping of those two gene versions with each other. Okay, so that would be the end results of the swapping. So if we did actually get the crossover between them, and notice that in that case you can get new combinations or those recombinant chromosomes. So based on this concept, geneticists have come up with what we call recombination frequencies. And recombination frequencies are frequencies that we can calculate that will tell us how likely, or you could say how frequently, does a crossover event occur between two specific genes. So in other words, how likely are we to get crossing over between this A and B, like we saw up here? And we could look at other genes. We could look at a combination of, if we had also on this particular chromosome, if I just add another gene here, what if we had down here at the bottom, we had a C? So if we had C, we could talk about crossing over between A and C, or B and C. So we're always looking at a pair of genes when we talk about these recombination frequencies. Now the key to understand here is that the greater the separation, so the greater the distance between two genes, the higher the recombination frequency. In other words, the more likely that there's going to be a crossover event between them. So if you have two genes that are located very far apart, then it's a good chance, or there is a good chance, that there will be crossing over between them. However, if you have genes that are located very close together, that recombination frequency, or the likelihood, is going to go down a lot, because there is just not as great of a distance for that particular event to occur within.